Hello student, welcome to EPG Pathsala. I am Dr. Ajit Jaswal from Department of Anthropology, Pondicherry Central University, Puducherry. Today we are going to discuss about a module entitled Branches of Human Genetics under paper Physical and Biological Anthropology. In this module, we are going to discuss about human genetics and emergence of its branches. We also try to understand the rationale of the technique in understanding the abnormalities and also trying to know about genetic diseases and its management. Let me give you a introductory information related to that. As we all know that biological anthropology is defined as the scientific study of humankind from the biocultural perspective. In understanding the human evolution and variation, biological anthropologists are interested in the area of human genetics to understand the origin and distribution of abnormal genes that causing certain abnormalities across the population group. Thus, human genetics provide wider scope for biological anthropologists to understand the concept of evolution of certain diseases and variation in the prevalence rate in the population segment. Human genetics is the scientific study of inherited human variation. In other words, we can say that human genetics is the ultimate answer in better understanding of us. Now let's define what is human genetics. Human genetics is actually a multidisciplinary field that connects the science of genetics and the growing number of people who can benefit from its application. It is the science of heredity, that is transmission of character from the parent to the offspring, which attempt to explain the similarities and dissimilarities among the related individuals. Recent advances in human genetics project have set free and in unending foundation of science, especially medical and therapeutic discoveries. They try to understand the field of human genetics in totality or in toto. We need to expose to various overlapping fields in human genetics like classical genetics, population genetics, cytogenetics, behavioral genetics, molecular genetics, biochemical genetics, and epigenetics. Classical genetics. Classical genetics is the branch of genetics deals with the analysis of parents to offspring. It depends on visible result of characters transmitted from parents to offspring. Mendelian experiments are made it possible to identify the basic mechanism of heredity. Cell division involving chromosome, which consist units of inheritance called gene or factors, the factors are the key component in classical genetics. Human cell contain 46 chromosomes, that is 22 pair of autosome and 2 sex chromosome. It may be XX or XY. Meiosis in germ cell produce 2 gametes, each with the haploid number of chromosome 23. Thus, offspring will receive 23 chromosomes from father and 23 chromosomes from mother. These chromosomes comply with the Mendelian's law, that is, first law is law of segregation. The gene pair or allele segregates during meiosis and the resultant gametes share each one allele. Second law says that the principle of law of independent assortment. Genes from different traits assort independently of one another. Later, several modifications have been made to Mendel's law. Because closely linked genes on same chromosome do not assort independently, due to crossing over may result in new combination of gene and in non-disjunction, daughter cell may receive both copies of information. This figure shows the inheritance of ABO blood group genes is the best example for classical genetics. Segregation of genes to gametes. For example, the probability that a gamete will receive the A allele is 0.5 from the law of segregation, and probability that this same gamete will receive the B allele is similarly 0.5. Thus, probability that a gamete will have a combined AB genotype is 0.5 into 0.5 equal to 0.25. The same probabilities are obtained for all four possible allele combinations, that is capital A capital B, small b, capital A small b, small a capital B. Since the number of gametes produced by an individual is very large, this probability translates directly into the frequencies at which each gamete type is actually present, and in turn the frequency with which each will be transmitted to offspring. Instead of normal segregation, non-disjunction may occur, although it is rare. The figure shows the process of non-disjunction in ABO system. RH blood group system is the best example. Capital D, capital C, capital E small e loci are closely linked on a chromosome. Hence, crossing over does not occur. Inheritance in the RH system, see figure. Because crossing over cannot occur, the genotype capital C small e or small d small c small e is impossible in a child from this mating. Even though the father has capital C small d and small e gene, the capital C small d e and small c small d small e on each chromosome are always transmitted to offspring together. In transfusion medicine, these principles are applied to identify the hemolytic disease of newborn. Currently, the science of genetics include a broad spectrum of inquiry from molecular studies on gene regulation to analysis of allele frequency in natural population. Now let's discuss about the population genetics. Population genetics is actually the study of frequency of allele in a population, means frequency of allele in the number of allele in a population. The discipline grew out of the need to establish Darwinian theory of evolution by natural selection on Mendelian hereditary principles. So, there is a Darwinian theory as well as modern Mendelian theory. Mendelian principle of inheritance, which is population genetics, are crucial to follow the Darwinian hypothesis that natural selection will play a very significant role in evolution and generating a diversity of life. Early population genetics like R.F. Fisher, J.B.S. Halden, Civil Wright, they used a single focus algebraic model to describe the changes in gene frequency at a particular level. Population genetics study the origin of variation, transmission of variant from a parent to the offspring generation after several generations, and the temporal changes that occur in a population because of the systematic and random evolutionary forces. In the year 1908, Hardy, a British mathematician, and Winberg, a German medical doctor, independently they formulated a theory of population genetics that is popularly known as Hardy-Weinberg law or law of equilibrium. This law says that allow us to predict a particular genotype frequency from its allele frequency. The law states that allele frequency remain constant throughout generation in a large random mating population, provided if the population is free from few important conditions: free from mutation, free from migration, free from genetic drift, free from natural selection. This allow quantifiable analysis of changes, and the equation can be extended to predict the changes in allele frequency resulting from continuing those changes. Thus, evolutionary outcome of a particular scenario can be predicted. The theory of population genetics is the theory of allele frequency. Each gene is in the genome exists in different allele states, called allele is an alternative form of gene. So, a diploid individual is either homozygote or heterozygote, similar or dissimilar. Within the population of individual, we can calculate the frequency of homozygote and frequency of heterozygotes of a gene. And from this, we can estimate the frequency of each allele. Let's suppose that in a population, a particular gene is segregating two alleles, capital A, small a, and that the frequency of A is denoted by small p and that of B is small a is denoted by q. Under this assumption that the members of the population mate randomly, then the diploid genotype of the next generation will be found by random union of haploid eggs and haploid sperm. The probability that an egg or sperm carries A is small p and the probability that it carries a small a is q. The probability of producing homozygote is p into p is equal to p square and the probability of producing a homozygote is q into q is q square. For capital A small a heterozygotes, there are two possibilities. First, A can unite with small a or small a can unite with capital A. Second, each of these occur with probability of p and q. Third, A and they are equally likely, the probability is 2pq. The predicted frequency are the term in the expansion of the binomial expression p plus q square is equal to p square plus q square plus 2pq and termed as Hardy-Weinberg genotype frequencies. Thus, Hardy and Weinberg principle demonstrated that given the assumption above, after one generation of random mating, stable genotype frequency will result and be maintained provided if the population is free from selection, mutation, migration or random drift, then variation will be preserved in a population. Cytogenetics. Cytogenetics deals with the physical basis of heredity. It can be termed as the study of chromosome, which are the visible carriers of the hereditary material. Cytogenetics describes the chromosome structure and identifies the abnormalities related to disease. The scope of cytogenetics includes several technologies besides FISH, FISH, what is also called as fluorescence in situ hybridization, CGH, cooperative genomic hybridization, multicolor FISH, etc. FISH remain the single most important technology in cytogenetics. Several innovations are described, of which the most important are single copy FISH, that is imaging of nucleic acid in living cell, and nanotechnology based FISH. The unique character of peptide nucleic acid P and A allows this proved to hybridize to target nucleic acid molecules more rapidly and with the higher affinity and specificity compared with DNA probe. PNA FISH is more suited for rapid diagnosis of infection. Chromosomal aberration can be detected based on microarray or biochip based technology, whose genome expressions array and direct molecular analysis without amplification are other important technologies. Analysis of single cell gene expression promises a more precise understanding of human disease pathogenesis and has important diagnostic application. Optical mapping can survey entire human genomes for insertion or deletion, which account for significantly greater proportion of genetic variation between closely related genomes as compared to single nucleotide polymorphism, and are a major cause of gene defects. Nanotechnology has facilitated the development of technology for single molecule imaging. Atomic force microscope has become a well-established technique for imaging single biomolecules under physiological conditions. The scanning probe microscope system emerged as an increasingly important tool for non-intrusive interrogation of biomolecular systems in vitro and has been applied to improve fish. There are connections between cytogenetics and biomarkers of genetics disorder as well as cancer. Biomarkers are very important for molecular diagnosis as a means to understand the phenomena of disease and as link between diagnostic and therapeutics. Biomarkers will play a major role in personalized medicine. Application of cytogenetics extend beyond genetic disorder and cancer to diagnose of several other diseases. Behavioral genetics. Behavioral genetics is a field in which genetic methodologies from twin and adoption studies through DNA will be applied to understand the individual differences in behavioral traits. It studies the inheritance of behavioral traits. In this field, emphasis will be given to understand how important are genetic and or environment influence on certain human behavioral trait or gene-environmental interaction. A strong correlation for certain behavioral trait found in parent and offspring to think whether it is parenting or gene. The science of behavioral genetics focuses to distinguish the effect of environment and genes. Human behavioral genetics use several methodologies to address the nature and mechanism of genetic influence on behavior. Twin studies and adoption studies are best examples to understand the importance of genetics and environmental effect. Heritability estimates the proportion of phenotypic variation in a population that is attributed to genetic influence. Heritability is a characteristic of population and not of an individual. Stature estimate is the best example of heritability. One individual may experience nutritional deficiency during development and keep shorter on the other hand. The other one is free from nutritional deficiency and attained full length. For the first, one environment is important, but the later one, genes are important. The level of genes and environment varies considerably at the level of individual within a population. Twin studies are much helpful in understanding gene-nutrition interaction. Identical, called as monozygotic twins, are alike in their genetic information. On the other hand, fraternal or dizygotic twins, though they are alike, share only half of their genes. Both kinds of twin exposed to similar degree of environment. The differential share of DNA in identical and fraternal twin is an evidence for a shared environmental contribution to the trait. The studies of children who have adopted at an early age provide an opportunity to segregate genetic effect from shared environmental effect. Molecular genetics. Molecular genetics deals with the chemical basis of heredity. It is the advanced field of genetics. Genomics and proteomics are the key component in molecular genetics. During the last half century, geneticists have determined the molecular structure of Mendel's factor and the mechanism by which they control the characteristics of an organism. Molecular genetics allow us to understand the DNA structure, nucleotide, DNA polynucleotide, the double helix, complementary base pairing, RNA structure and interrelated relation between the DNA and RNA molecules. Molecular genetic research has given us powerful tool to study genes. In 1985, Carey Mullis developed a technique called PCR, polymerase chain reaction, that allow us to take a minute sample of DNA and produce millions of copies of DNA for molecular analysis. With the use of PCR, DNA isolated from a small sample of tissue, perhaps a single sperm or white blood cell or a hair follicle recovered from any crime scene, can be subjected to molecular analysis. Forensic scientists use a type of DNA analysis called DNA fingerprinting to examine the variation in the sequence of DNA. A major component of DNA technology is cloning, which is the process of making multiple identical copy of a gene by incorporating in an organism like bacteria to use in different applications. DNA recombinant technology allowed us to alter the DNA with the help of restriction end nuclease and can be inserted into another matched DNA to understand the variable pattern. This technique helpful in merging the gene into molecular device and can transport these genes to various cellular destinations called gene therapy. In this technique, corrected gene can be delivered into individuals with defective gene that cause specific disease. DNA chip technology facilitates gene expression profiling to understand the specific gene up and down regulation. The up and down regulation of gene allow us to estimate the cancer risk. Molecular genetics has revolutionized biology and is having an ever-increasing impact in pharmacology, genetic engineering and disease prevention, in increasing agricultural growth, in detecting of disease and crime, that is forensic, etc. Further, it helps us in tracking DNA evolution, migratory pattern and species evolution over the ages. The advance brought in molecular genetics have profound implication for the future of medicine, for they had placed us at the threshold of new method of diagnosis, prevention and treatment of numerous human diseases. Hormones, vaccines, therapeutic agents and diagnostic tools are already greatly enhancing medical practices. Now let's discuss about the human biochemical genetics. Biochemical genetics is an important branch of human genetics, which provide a base to understand the genetic variation and disease occurrence. Human biochemical genetics expose how the study of human system led to advances in basic biology. Recent development in the whole human genome project has been a driving force for identifying genetic disorder. This is true in the area of inborn error of metabolism. From the Gerard's understanding of Alkaptonuria to today's ability to detect single nucleotide changes, we have a technology driven discoveries. Various technologies like amino acid analyzer, gas chromatography or mass spectrometer led to identify the analysis of amino acid, organic acid and fatty acid. Identification of these disorders at an early stage may provide an opportunity to initiate therapies like dietary modification, enzyme inhibitors, enzyme replacement therapy and bone marrow transplantation. Let's discuss about amino acid metabolism disorder. Phenylketonuria is a rare genetic disorder where body cannot break down the essential amino acid phenylalanine. In the body, phenyl hydroxylase enzyme convert phenylalanine to tyrosine. Tyrosine is necessary to activate neurotransmitters like epinephrine, norepinephrine and dopamine. The increased level of phenylalanine in the blood cause damage to the nervous system and brain. Phenylketonuria is inherited as autosomal recessive pattern. An infant with the phenylketonuria may appear normal for first few months of his or her life. If the condition is not treated in time, abnormalities like mental retardation, jerky hand and leg movement, hyperactivity and stunted growth will take place. Next is carbohydrate disorder. Galactosemia is a disorder that affects how the body processes a simple sugar called galactose. The signs and symptoms of galactosemia result from inability to use galactose to produce energy and affect different enzymes involved in the breaking down the galactose. If the condition is not addressed with low galactose diet, severe complication may arise few days after birth. This condition is inherited as autosomal recessive pattern. Fatty acid oxidation disorder. Medium chain acyl coenzyme A dehydrogenase is a condition when body fails to convert certain fats to energy during starvation or fasting. MCAD may include the symptom of vomiting, lethargy and low blood sugar. The condition is inherited as autosomal recessive pattern. Next one is organic acid disorder. Isovaleric acidemia is a rare disorder in which body is unable to convert certain protein properly and help to build up of a particular acid known as organic acid. Abnormal level of organic acid in the blood, urine and tissue can be toxic and can result in serious health problems. The condition is inherited as autosomal recessive pattern. Similarly, we'll see the lysosomal storage disease, mitochondrial disorder and urea cycle disorder. Epigenetics. Epigenetics involves change in phenotype without change in genotype. It can also be referred to as change in organism, caused by alteration in gene expression rather than alteration in genetic code or DNA. Epigenetic changes will be influenced by several factors such as age, lifestyle and disease. Epigenetic changes will be more rampant than can end up with disease like cancer. The term epigenetics was first introduced by Waddington in 1942 and described the influence of genetic processes on development. The concept of genetic assimilation has gained interest during 1990s, which paved the way to understand the molecular basis of Waddington's observation. in which environmental stress causes genetic assimilation of certain phenotypic characters in Drosophila fruit flies. Thereafter, efforts towards identifying epigenetic mechanisms related to external factors has been attenuated. DNA methylation, histone modification, and non-coding RNA-associated gene silencing are currently considered to be important in initiating constant epigenetic changes. The science of epigenetics is quickly growing. By understanding both the environment and the lifestyle, which may directly interact with genome to influence epigenetic change. This change may precipitate any stage of human journey and may see in later generation also. Prenatal and postnatal environmental factors influence the adult risk of developing various chronic diseases. Research results shows that starvation in early stage or poor nourishment in the womb may result in obesity and hypertension in later life. A few epigenetic mediated disorders are mentioned below. A few epigenetic mediated disorders are cancer, immunity, neuropsychiatric disorder, pediatric syndromes, mental retardation disorder. The the scientists observed that genes of colorectal cancer cell substantially hypomethylated compared with normal tissue, which can activate oncogene and initiate chromosome instability, whereas DNA hypermethylation initiates silencing of tumor suppressor gene. Addition of genetic and epigenetic error can transform a normal cell into metastatic tumor cell. Similar pattern of changes can be seen in other cancer also. Hence, epigenetic changes can also be used as a biomarker for early diagnosis of cancer. Okay, now let's try to summarize the whole information that we have discussed in this topic with certain important points. As human genetics provides a wider scope for biological anthropologists to understand the evolution of a certain disease and variation in the prevalence rate in population segment. Human genetics is the scientific study of inherited human variation. Hence, there is a need for anthropologists to learn about human genetics to answer several questions which continue to be under investigation towards the human welfare. To understand the field of human genetics, it is very important that we need to expose to various overlapping fields in human genetics like classical genetics, population genetics, cytogenetics, behavioral genetics, molecular genetics, biochemical genetics, and epigenetics. In all these specialties, newer technology has been developed and continue to be under investigation to provide more and more sophisticated tool with which we can understand evolutionary pattern of variation and disease occurrence in different population. Thank you.